I am Dr. Sagar Majumder, Assistant Professor of Department of Biochemistry. So, I am going to teach you the biochemistry part. This is chemistry as such is not that of interesting part for me, but many of you may be interesting, maybe it may be interesting for you, but we have to learn it because in a given fraction of, in a given moment, there are thousands of biochemical reaction is going on because of which you are alive. Okay? So this is very important. Any deviation from this normal biochemical reaction will make you sick. And when there is a defect in the biochemical reaction in the body, surely there will be some abnormal molecule will be, biochemical molecule will be there in the blood or body fluid. So we are going to diagnose the disease by identifying that biochemical molecule in the blood, and we are going to determine what is the disease. Okay? When you find, you might have seen, when one of your relative is sick, a doctor is writing, say, 10 numbers of investigation. Okay? Out of that, at least 7 will be biochemistry related investigation. Okay? In every subject, every discipline of medical science is based on biochemistry. It is the biochemistry where enormous research advance has been occurred, and it is the subject which is directly helping the medical profession to diagnose, to treat the patients. So, very important, even though it is not that interesting because you cannot see the thing, but you have to make yourself in such a way that you can remember the thing. So what is that way? You try to write the thing, whatever you learn, write it down. Okay? Make some diagrams, make some flow charts, then only you can remember it. Once you have read the thing, you will remember only 10 to 20%. Okay? You have to read it again. Then you will be able to remembering the thing. Okay? Now, let me know who is roll number 1. Roll number 1. Yeah, what is your name? Pardon? You are from Tripura, is it? Okay. Now, you are from Bengali medium or English medium? Okay. So many of you may be from local language of education, and to be in the medical profession, it doesn't matter. Okay? For us also, we are from the Bengali medium, so we could learn this medicine without any problem. In the beginning, say 1.5 months, you may feel a bit difficult, but as because it is not a literature, only few words, should I say, will be repeatedly used. So that will not be a problem. You already know the thing. Session, you know. So it's very easy to get drowsy. Huh? But biochemistry again, it is both. So something we must do so that we don't feel sleepy. Huh? Well, so we'll proceed for today's topics. Now, before we go, I tell you one thing, your attendance is strictly observed in medical science. Okay? Whatsoever good student you are, if you do not have the prescribed attendance, percentage of attendance, you will not be allowed to appear examination, your final examination. In theory, you must have 75% attendance, and in practical 80% attendance. Okay? Number 2, try to be in time. Try to enter the class within time. Okay? Otherwise, say again and again disturbing means we are also feel disturbed. No, there is a break in the lecture process. So try to come in class within the prescribed time, and up to 5 minutes you will be allowed. After that, you may not be given. You will be allowed to enter, but may not be given attendance. So almost every week you will have examination. Examination is assessment system. Okay. Otherwise, is very easy to get lazy. You know. So if you don't finish up the thing what is taught today, you should read at least once today itself, so that you can remember. We will go through the basic things which you must know. After that, if you want to know more, you can adapt from the books. What book you have purchased? Vasudevan, Satyanarayan. Okay, Satyanarayan is a easy book. You can follow that. Easy language, easy to understand. Vasudevan is very good book, but so many informations are there. You may not require so many things for your undergraduate level, but it's a very good book. If you can follow, that's nice. The another book has come, very easy language and very good book. It is S.K. Datta. That is also very good book. Okay. Follow one. Whatever you follow the one book only. Satyanarayan is good. You follow one book. Okay. And try to read from that book. If you want to refer to other book, you can take as reference any extra information you note in your Satyanarayan, so that you don't have to flip all books during examination. That time it will be very difficult. So extra things whatever you want to note, you just note down in the Satyanarayan. Okay. For example, so one book you follow, in that any additional information you can add. So we'll start with carbohydrate chemistry. Have you heard of carbohydrate? Just give one example. Carbohydrate, glucose, sucrose, maltose. Okay. Today you have taken food. Okay. You have taken plenty of carbohydrate. No? But whatever you have taken, whatever it has gone in your stomach will not be absorbed. That will be simplified into glucose, and then only it will get absorbed. And it is the glucose which is the most important simple carbohydrate, which is biologically very important. Okay. This is your competency 3.1. You must know definition. Yeah. The competency says discuss and differentiate monosaccharide, disaccharide, polysaccharide with examples. Their importance as energy fuel, structural element, and storage molecule in the body. In this competency, you have to know this. What is monosaccharide? What is disaccharide? What is polysaccharide? With example, their importance and how these carbohydrate or these monosaccharide are stored in the body as polysaccharide. So, you must know the definition of carbohydrate, function, classification with example, sorry, properties of monosaccharide, though in competency properties are not mentioned, but you have to know the properties. In practical class, you will do some experiment, then in the examination you will be asked about that experiment. There you will be asked some questions which will cover from which is from monosaccharide these properties. Okay, then chemical properties. So carbohydrate, this term is derived from their general formula CH2O, and that takes them seems to be hydrates of carbon. Carbohydrate often we term as saccharides, say monosaccharide, disaccharide. Okay, they are closely related, these terms. Most abundant carbohydrates are polysaccharide. Today also when you have taken food, you have taken rice. In the dish, it occupied the maximum part of the dish. Okay? So a lot of carbohydrate you have taken. In our dish, predominantly we eat carbohydrate with small amount of protein and fat. Most important carbohydrate in the body is glucose. Whatsoever the form polysaccharide, disaccharide, whatever you take, it will be utilized in the body as glucose. And it is the glucose which take part in different form for the formation of other biomolecules. Very important. Each time we ask, what do you understand by carbohydrate, or what is the definition of carbohydrate? Okay, but unfortunately, maximum of the students they fumble. So there should not be any confusion. No, confusion should not be there. So carbohydrate may be defined as the polyhydroxyaldehyde or ketones or compound that yield them on hydrolysis. Okay. Now polyhydroxy you can see. So see, so many hydroxyl groups are there. So it is polyhydroxyaldehyde or ketones. Polyhydroxyaldehyde or ketone or any compound after hydrolysis produce this. They are termed as carbohydrate. What is the function of carbohydrate? Can you tell me one? You can see from here itself, energy. Energy, glucose is the, carbohydrate is the energy providing food. Protein is the, what type of food? And fat is, okay, fat is also, it also provides energy, but it has to convert into this one, carbohydrate, a part. So energy is provided from glucose. Each gram of glucose provides 4 kilocalories of energy. Intermediate of metabolism. When you take carbohydrate, it is metabolized and gives carbohydrate. When you take protein, it is metabolized and gives carbohydrate also, some other compound also. Okay. Now, in case of metabolism, the one or more intermediate will be formed, which is carbohydrate. and from there some other thing may be synthesized. Okay, but there is definitely synthesis of production of the carbohydrate. Then storage form. Carbohydrate means, if we take the example of glucose, in animal it is stored as glycogen. Around 400 grams of glycogens are stored in the body as fuel reserve. This glycogen, when energy is immediately required, it is converted into glucose, and glucose provides energy. Okay. What is the energy currency of body? Very good. It is ATP. Okay. ATP is synthesized through TCA cycle, oxidative phosphorylation, electron transport chain into ATP. That you will know afterwards. Structural components. This is very important. Say plasma membrane. There is lipid bilayer where surface glycoproteins are there. Okay, so they are taking part in structural protein. Sialic acid, which is also a kind of carbohydrate, is taking part in structural protein. Okay, then chitins. Okay, when we discuss polysaccharide, you will come to know. Fibers. In animal, it is the glycogen which is stored as stored energy. In case of plant, it is what? Any idea? Starch. It is stored as starch. Okay. Now another thing you have come to know, cellulose. No, cellulose. Have you heard of cellulose? Cellulose also, it is a polymer of glucose. Okay. So that is also is a kind of fiber. Okay, fiber. So there are so many dietary fibers are there. Some fibers are soluble fibers. They play very important role in maintaining body health. Say fibers, they provide stool consistency into soft stool, so that easily it can be excreted out. They prevent toxic material from getting absorbed in the body. Okay, so there are so many functions are there. We will come to know afterwards. Then constituents of nucleotides. Nucleotides. Have you got in your? Have you heard in your class 12 standard? You heard nucleotides. One nucleotide. One nucleotide, adenine. Okay. Now in adenine, there is ribose. Ribose is there, deoxyribose. In DNA, there is deoxyribose. In RNA, there is ribose. Okay, all are carbohydrates only. Okay, pentoses. Detoxification. So detoxification, when you take food, a plenty of toxic material is taken along with the food. Whatever we take, everything is good, it's not there. There are some alkaloids, there are so many things in the food which is being absorbed, which is being taken by the human being and absorbed in the body, and that is eliminated by detoxification by the liver. Okay. Now, one thing I will tell you, in the body endogenously produced bilirubin. Bilirubin is produced. It is toxic to the body. So, in the liver, what happens? A glucuronic acid is conjugated with the bilirubin, and bilirubin is excreted by conjugating with the glucuronide. So, this glucuronide is nothing but a carbohydrate. Okay. So, these are the some important functions, and some more are there in book, you can add up. So here we will just remember some terms. Aldose. Aldose, a carbohydrate that contains aldehyde group. Here it is aldehyde group. So when this group is there in the carbohydrate, we will term them as aldose. Ketose. When a keto group is there, we will term them as ketose. Triose. We have seen 3 carbon compound. We have seen here 6 carbon compound glucose. So when there is 6 carbon, we tell them as hexose. Okay. When there is 3 carbon compound, we will term them as triose. When 4, tetrose. Likewise. Okay. So to classify, in maximum of the book you will find there are three types of carbohydrate: monosaccharide, oligosaccharide and polysaccharide. Okay, but examiners, they sometimes again ask what is disaccharide. Okay, so in some books they have mentioned monosaccharide, disaccharide, oligosaccharide, polysaccharide. So when you also mention 4: monosaccharide, disaccharide, oligosaccharide, polysaccharide. Monosaccharide, when there is one sugar moiety, simplest sugar moiety in the given carbohydrate, we tell is a monosaccharide. So sugar, you also know sugar, but I am also telling sugar. What is the sugar? What you know? It is the table sugar. Okay. When we tell table sugar in our language as biochemistry, we tell it as sucrose. Okay. Now, when teaching the thing, when we pronounce it sugar, you take it as carbohydrate or monosaccharide. Okay. So when there is only one sugar moiety is there, that is called monosaccharide. When there are 2, it is called disaccharide. And oligosaccharide is from the 3 to 10 numbers of sugar moiety is there. When there is 3 to 10 numbers of sugar moiety is there, that is called oligosaccharide. But in Satyanarayan also, they have mentioned 3: monosaccharide, oligosaccharide and polysaccharide. Accordingly, they have defined oligosaccharide as moiety having carbohydrate having 2 to 10 numbers of simple sugar moiety. Okay. So if you say oligosaccharide 3 division, monosaccharide, oligosaccharide and polysaccharide, then oligosaccharide will have 2 to 10. But when you divide into 4, so disaccharide is getting separated, so you tell 3 to 10 oligosaccharide. Okay. Polysaccharide, it is above 10. Above 10 numbers of glucose, when it is there, we call it as polysaccharide. So far, what is carbohydrate? Tell me, what is carbohydrate? Polyhydroxy, aldehyde or ketones or any substance which give them this polyhydroxy ketones or aldehyde on hydrolysis. Okay, that are called carbohydrates. How many types of carbohydrates are there? 4. Or if you like to say 3, that is also acceptable. 4, I will say monosaccharide, disaccharide, oligosaccharide and polysaccharide. What is oligosaccharide? 3 to 10. According to this slide, 3 to 10. If you say 3, then it will be 2 to 10. Understood now? This part, no confusion, everybody? Okay. So let oligosaccharide part and polysaccharide part we will discuss later on. We will go for the monosaccharide. So in classification, depending on the number of simple sugar, we divide into mono, di, oligo and polysaccharide. Okay. Now monosaccharide again divided into two. Aldose and ketose. Aldoses and ketoses, disaccharide into two, reducing disaccharide, non-reducing disaccharide. Oligosaccharide, there is no division. Polysaccharide, homopolysaccharide, heteropolysaccharide. When there is only same type of monosaccharide is there, that is called homopolysaccharide. When there is different type of monosaccharides are there in the polysaccharide chain, that is called heteropolysaccharide. Okay. Now homopolysaccharide, it may be branched or unbranched. Okay. Example already here given. Aldoses, glucose, ketoses, fructose, reducing disaccharides, maltose, non-reducing sucrose. Okay. And of these things you will know when you go for polysaccharide. Now these are the number, depending on the number of simple sugar present in the polysaccharide carbohydrate. Now it is the depending on the monosaccharide is divided into two, depending on the functional group, aldehyde group and keto group. Now another division is there, depending on the number of carbon atoms. Number of carbon atoms in the monosaccharide. When there is 3, there this is called triose. When there is 4, tetrose. When there is 5, pentoses. Hexoses. Heptoses. Okay. Now these are the numbers of carbon. It has 3 carbon, it has 4 carbon, it has 5 carbon, 6 carbon, 7 carbon. Now a aldose having 3 carbon is called aldotriose. Likewise, aldotetrose. Aldopentose, aldohexose, aldoheptose. Having 3 carbon, having keto group, this is called keto triose. So like that, keto triose, keto hexose, tetrose, keto pentose, keto hexose, keto heptose. Okay. Now Example, aldotriose glyceraldehyde. Aldotriose is glyceraldehyde, very, very important molecule. Glyceraldehyde. Aldopentose is dihydroxyacetone. This is also again important molecule that you will come when you go for this one metabolism part. Then aldohexose. Aldohexose, glucose, galactose. Ketohexose, fructose, sorbose. Okay. Now important is fructose. Aldohexose, manohexose. Ketohexose is pseudohexulose. So when there is metabolism of different molecules, this will come as a intermediary product. You will know it. Okay. So like that, they are classified. Monosaccharide. To go a bit detail in monosaccharide, mono means one, saccharide means sugar. So there is one sugar moiety means one simplest sugar. When carbohydrate is extremely hydrolyzed and cannot be further hydrolyzed, that is called monosaccharide. Sorry. So as you told, there are aldoses and ketoses. Depending upon the number of atoms, they are triose, tetrose, pentose, hexose, heptose. Here are some examples. You will get it in Satyanaরায়ণ also. Every book it is there. Okay. Now, where these monosaccharides are found, they have mentioned here. This part we will discuss later on. Now, structural functional relationship of monosaccharide. Glucose is the most important physiological and biochemical monosaccharide. Can be represented, each structure can be represented as straight chain structure or ring structure or boat and chair form. So, first two are very important. This is a straight chain structure. Aldehyde group, terminal alcohol group, carbon number 1, 2, 3, 4, 5. Okay. Now, there are multiple hydroxyl groups. These hydroxyl groups, they can interchange with each other. They can change their side. Okay. With aldehyde of monosaccharide, when alcohol is reacted, they form hemiacetal. And with ketone group, when alcohol is reacted, they form hemiketal. These two terms you remember. Isomerism. You will get a short note on isomerism of monosaccharide. Okay. The compound possessing same molecular formula and have different structures are referred to as isomers. Okay. See this glucose. Glucose is having C6O12O6. Fructose also is having formula C6H12O6. But there is a difference. No? There is difference. The first carbon of the glucose is having aldehyde group. Second carbon of the fructose is having keto group. So even though formula is same, their groups are different. That's why they are called structural isomers. Okay, isomerism, and the process of the phenomenon of existence of the isomer is called isomerism. Another is stereoisomer. So what I told in sugar, glucose and fructose, it is the aldose ketose isomerism. See, this is the keto group, this is the aldehyde group. So, it is the aldose ketose isomerism. Stereoisomerism, same molecular formula, but structures differ in their configuration, means there is spatial difference in spatial arrangement of the groups. Because of the asymmetric carbon, this stereoisomerism it occurs. Okay. Now, asymmetric carbon, what is that? Asymmetric carbon or chiral carbon. Chiral carbon. When 4 different atoms or molecules are attached in one carbon, that carbon is called asymmetric carbon or chiral carbon. Okay. Now, it is important because when these 4 atoms, they can change the side. Okay. Now, see, this is the triose, simplest triose. So, it has a carbon here having 4 different atoms attached to that. Okay. This is alcohol group, this is hydroxyl group, this is hydrogen, this is carbon. So, this glyceraldehyde has one asymmetric carbon, and that is responsible for isomerism. See, the hydroxyl group here is in the right side and hydroxyl group here is left side. While this group, hydroxyl group, is attached in the right side, these are called dextro type of isomer, and this is levo type of isomer. Okay, this is in the left side. So, these are the structural isomers due to the anomeric asymmetric carbon or chiral carbon. So, now when in this glyceraldehyde, it is taken as reference molecule. Now, if we go on adding the carbon molecule here, it will gain its length, and likewise a glucose can give synthesized, and where this hydroxyl group, when it is attached to the left, it will to the right, it is called D-glucose, and when it is in the left, it is called L-glucose. In nature, it is the D-glucose which is available in plenty, and body can metabolize only the D-glucose. Okay? The enzyme responsible for metabolism of glucose can metabolize only the D-glucose. Okay? That is why it is biologically important molecule. DL isomerism, when it is just mirror image form, this is called enantiomers. Enantiomers. Just see, here is a mirror and it is just opposite to that. Now, this group we can rewrite as like that, so that there will not be any confusion. Just last part. C H H O H. Last part, if we write like that, this part, it is just like a mirror image. So, this is the enantiomers. They are called enantiomers. Another term is there, optical isomerism. Optical isomers. When plain polarized light is thrown in a solution containing B or this glucose solution, they can rotate the polarized light towards the left or right. When light When the light is rotated to the right, that means clockwise, they are termed as dextrorotatory. It is denoted by D, small d or plus. When light is rotated to the left or anti-clockwise direction, the substance is said to be levorotatory type. When equal amount of D and L isomers are present in the mixture, they are called racemic mixture. And in such situation, in racemic mixture, there is no rotation of the polarized light. This is a simple diagram of polarimeter. So light is thrown for here. This is unpolarized light. Then one filter is there and it polarizes the light like that. Then this is the tube containing solution, and when this light is passed through this, there is a rotation. See, gradual rotation is there. So there is rotation of the light. Here there is no rotation. When it passes through the solution, there is rotation. So from eye, if you see which side the rotation is there, the person see that there is a rotation of light towards the clock. Okay, so this is dextrorotatory. Epimerism. When a functional group is attached in one carbon in a right side or left side comparing to the glucose, that is called epimer. Here, see, this is the glucose. Now, this is galactose. All are C6H12O6. Here in number, carbon number 4, 1, 2, 3, 4, carbon number 4, in relation to carbon number 4 of D-glucose. Why it is D? Because this fifth carbon, it is having hydroxyl group towards the right. So that's why it is D-glucose. Now, in a fourth carbon, the hydroxyl group is in the right side, but in the fourth carbon of galactose, hydroxyl group is in left side. Fifth carbon hydroxyl group is in the right side, so D-galactose. Here also D-glucose. Here also D-mannose. But in fourth carbon, there is change of this attachment of the hydroxyl group. That's why galactose and glucose are epimers. Okay, epimers in relation to fourth carbon. And mannose, it is the epimers in relation to the second carbon. Okay. Now this is the epimer of glucose in relation to second carbon. This is the epimer of glucose in relation to fourth carbon. So what is the relation between galactose and mannose? There is some relation of galactose in this one, galactose and mannose, no? And in this situation, these two molecules are called diastereoisomerism. Diastereoisomerism. There is a difference in attachment of hydroxyl group, but not in the same carbon. Okay, this is not in the same carbon. It is, one is in the carbon number C, carbon number 4, C in relation to carbon number 4, and another is carbon number 2, C. So in two different carbon, functional groups are attached differently. So they are called diastereoisomerism. Anomerism. Short note, anomeric carbon. Anomerism is when we put glucose in solution, they have a tendency to form a cycle, a cyclic structure. Okay, cyclic structure. In that case, the bond occurs between the first carbon, in case of glucose, aldehyde group, and the hydroxyl group of fifth carbon. In case of fructose, the keto group of second carbon and the fifth hydroxyl group of fifth carbon. Okay, they form a closed ring structure. Okay, that is called pyranose ring. Now here you see a ring structure is formed from the carbon 1 and carbon 5 hydroxyl group taken part in the formation of the ring structure. And here this carbon, which was not anomeric carbon earlier, has become a not anomeric, which was not chiral carbon earlier or asymmetric carbon earlier, now has become asymmetric carbon because of the formation of the ring structure. Okay. So this is called the anomeric carbon. The carbon atom or hemiacetal carbon atom of aldehyde group or the ketone group in cyclic form of the glucose monosaccharide structure becomes anomeric because of the formation of the ring structure. So this is called anomeric carbon, and it is anomeric carbon. It has a why it is important? Because in anomeric carbon, when hydroxyl group is attached towards the side of alcoholic group, then it is called alpha D glucose. Then in case of hydroxyl group, if it is opposite to the alcoholic group, then it is called beta D glucose. D because here both are in the hydroxyl group is in the right side. Mutation rotation, it is defined as change in the specific optical rotation representing interconversion of alpha and beta form of D-glucose. So because of the anomeric carbon, there is formation of alpha and beta anomers, and they provide specific rotation. In case of alpha D-glucose, it is 112, beta D-glucose, it is 18. When kept in mixture, there is spontaneous conversion of alpha to beta, beta to alpha. Okay. Now, in a stable solution, it becomes, the specific rotation becomes 52, because when it is kept, mixture is kept, 63% of the glucose solution having beta D glucose and only 36% is having alpha D glucose. So resultant, it is because beta D glucose is more stable than the alpha D glucose. So the resultant specific rotation becomes 52.7 plus 52.7, and 1% of the glucose, it remains as open chain. So what is mutation rotation? This is defined as the change in the specific optical rotation representing interconversion of alpha and beta form of D-glucose to an equilibrium mixture. So this is the interconversion. Chemical properties. These are important because of your practical class. Furfural formation. When hexose is treated with concentrated sulfuric acid, they 3 numbers of hydroxyl group is removed and hydroxymethyl furfural is formed. Ketoses, when mixed with concentrated sulfuric acid, they form furfural. This furfural with phenolic compound gives color compound. This is the principle of Molick's test for identification of this monosaccharide in oligosaccharide and polysaccharide. When oligosaccharide or polysaccharide is mixed with this acid, concentrated sulfuric acid, there is acid hydrolyzation of these polysaccharide and oligosaccharide. They yield glucose. Okay, so this glucose, it reacts with sulfuric acid and gets dehydrated by removal of hydroxyl group and furfural is formed. So this is the principle of the Molick's reaction. When glucose is kept in alkaline solution for several hours, it undergoes isomerization to form D-fructose, D-mannose. This results formation of common intermediate anadiol. This process of shifting of hydrogen atom from one carbon atom to another produces anadiol is known as tautomerization. Okay. Now this anadiol is very highly reactive. Okay. This is the anadiol. There is change of hydrogen ion from here to here, like that. There is spontaneous change. So there is conversion of fructose, mannose and glucose with an intermediate anadiol formation. Anadiols are good reducing agent and form basis of reducing property of sugar. Benedict test and Freling's test. You are going to do in practical Benedict test to identify the reducing sugar. A person suffering from diabetes mellitus passes glucose in the urine. So we identify the glucose by Benedict test. Okay. So Benedict test, what is the principle of Benedict test? Benedict test principle is, in alkaline medium, this glucose, it forms anadiol, and that anadiol reduces cupric ion to cuprous ion, which is red in color. So seeing that red color in the test tube, we identify how much sugar is present in his urine. Okay. Or whether sugar is present or not. From concentration of the color, we can identify what is the percentage of sugar in the urine, and that's why it is called semi-quantitative test that you will know in your practical class. Oxidation, there is aldehyde group, there is alcohol group, last alcohol group. So any of the group may be oxidized or both can be oxidized. So when aldehyde group is oxidized, this is called gluconic acid, and terminal alcohol group is when oxidized, it is called glucuronic acid. So these are the intermediary products, and they are required in different biological functions. Reduction, both aldoses may be reduced by enzyme corresponding polyhydroxy alcohol, like see sorbitol, mannitol, dalcitol, ribitol. Sorbitol many a time we use as stool softener, sorbitol. Then mannitol, many a time we use when there is head injury. To reduce the intracranial tension, we use mannitol. Then to increase the urine outflow, we use mannitol. Okay, so these are the having important use as drug. Mannitol is used as an osmotic diuretic, reduce the cerebral edema. Sorbitol, it accumulates in the lenses. Sorbitol, it is important because sorbitol, it accumulates in the lens and produces opacity of the lens. That is called diabetic cataract. Usazon formation, that is also again an important thing to identify monosaccharide. Presence of free carbonyl group and carbonyl ion, aldehyde or ketone group, what we have seen. These are essential for usazon formation. The first carbon is aldehyde in case of glucose, second carbon is ketone in case of fructose. Okay. Now carbon number 3, 4, 5, 6. Last four carbons are not unchanged. So when we add phenyl hydrazine, in the first carbon and second carbon, it gets attached. Okay. Now the first carbon having aldehyde group in case of glucose, the second carbon having keto group in fructose is merged. It becomes same. Rest of the 3, 4, 5, 6, these 4 carbons, they are equal in both the things. Okay. So both the glucose and fructose. So when usazone is formed, it appears same. Glucosazone, fructosazone, and C. Glucose. This is glucosazone. Glucosazone is it is involving the first and second carbon. It is the first reaction replacing this one hydrogen oxygen in the first carbon. Then in second reaction, it is replacing the hydroxyl group in the second carbon. So it is like that. Now if you see the rest of the carbons in fructose, glucose, fructose and mannose, these are same. So these glucose, fructose, mannose, they will give same type of usazone formation, broom shape. See, this is the needle shape usazone. In case of lactosazone, powder puff like. In case of maltosazone, the usazone is sunflower like. Glucoside formation. When a hydroxyl group on the anomeric carbon of a monosaccharide reacts with hydroxyl group of another carbohydrate or non-carbohydrate, leads to the glucoside formation. So first carbon of the glucose may react with the another carbon hydroxyl group of the another carbon. Okay. Now second carbon, it may not react with the carbon having functional group, that means first carbon, it remains open. So if one of the glucose moiety is having free aldehyde group or keto group, it will be reducing sugar even if it is disaccharide. Okay. Now when glucose and glucose is involved in glycosidic bond formation, it is called O-glucosidic bond. When glucosidic bond is between glucose and nitrogen atom of the other molecule, it is called N-glucosidic bond. Such type of bond is there in case of adenosine. Adenosine, say adenine nucleoside, adenosine nucleoside. Okay, it is nucleoside. So in case of adenine, there is nitrogen in the ring. That nitrogen reacts with the ribose carbon of the hydroxyl group of the ribose. So that's why here it is called N-glycosidic bond. See, this is the ring structure of glucose, pyranose ring. This is the number 1 carbon. This is the hydroxyl group of carbon 1, which is anomeric carbon, which gives the reducing property of this sugar, reacts with the 4th hydroxyl group attached to the 4th carbon of the second glucose. Here the first glucose is free to react as anomeric carbon. So this disaccharide is having O type of glycosides bond, and as the first carbon of the second glucose moiety is free, it is reducing. When we discuss disaccharide next class, we will tell you. Here is the N-glycosidic bond. Nitrogen is reacting with this carbon of the ribose. Physiologically important glycosides. Some of the glycosides are very important for medicinal purpose. Streptomycin. Streptomycin, it is an antibiotic. It is nothing but the glycoside, physiologically important glycoside. Then cardiac glycoside, digoxin. Streptomycin is used for treatment of tuberculosis. Still now it is used. Then cardiac glycoside like digoxin, digitoxin, they are used in congestive heart failure. Then anthracyclic glycoside, doxorubicin, is used for treatment of cancer. Then quabain, that is used for inhibition of sodium potassium ATPase channel. Some derivatives of monosaccharide, phosphoric acid derivative like glucose 1-phosphate, this is the intermediary molecule produced during metabolism. Amino sugars, glucosamine, we will read next class. Then deoxy sugars present in the DNA, sugar acid, glucuronic acid, then sugar alcohol, these all are required for the different production of different component of the in the body. So disaccharide and polysaccharide we will read in next class. So next class you read definition of carbohydrate, classification of carbohydrate, isomerism, anomerism, epimerism, okay, stereoisomerism. So this thing we will recapitulate, we will ask you, read the thing today itself, okay, thank you.